Hey guys, what is up? Welcome to the video. First of all, I want to say thank you so much for your support to the science series. This is a series for you guys, so thank you very much. Please leave a like, please subscribe and spread the word. The reason why I started this series was because I had a dream that I was doing this. Maybe this is a big person who's told me to do it in a dream, I don't know. But basically, when you guys return to school, if you do in September, you'll be in bubbles, which means you can't move around a lot. So I don't think you're going to learn the way you usually do. So that's why these series are important. Anyway boys, today we're looking at enzymes. Now enzymes are very, very important. When I was a kid, I was always fascinated with when I eat this food, what happens to it? We're going to find out today. Boys, first of all, I'd like you, and girls, sorry, ladies and gentlemen, I would like you to pause the video and see if you could name the different parts of the digestive system. You can pause it now. I'm not going to wait because, you know, time is of the essence. So you can pause the video. Here's the answers. Okay, we've got mouth. Now, I just want to go back one sec. Now, it's very important to understand one thing. The esophagus and trachea, it's a misconception. I used to get confused what's what. The esophagus leads to the stomach. However, the trachea leads to the lungs. So this actually goes down to the stomach, the pancreas, the gallbladder, the liver. Boys and girls, remember all of it. Okay? Hard to talk about digestion when you don't know what's what. Then when your important organs in the digestive system. Okay. So really our objective today is to describe the role of enzymes during digestion, and hopefully by the end of it you can name the main 3 enzymes. You can also tell me about the lock and key method, and if you're really smart you can tell me about peristalsis. Okay? Okay, so these are some of the keywords I'd like you to implement when the tasks come for you guys. It's very important to understand these keywords, and I'll be explaining them. Okay, first of all, let's go over this. Okay, enzymes are biological catalysts that speed up reactions without getting used up. They break up large molecules into smaller ones. Enzymes have a 3D structure. It's very important to understand these enzymes have a specific shape. Okay, the shape over there is called the active site. The substrate or the food product must completely fit inside the enzyme for the reaction to take place. Okay, this is exactly what I'm talking about. If you guys can see, this substrate or food product fits perfectly inside there. If it didn't, it wouldn't work. So you need to understand that once they come together, then the enzyme will break the substrate down into products. Okay? So each enzyme is specific to a particular substrate that are similar in structure. Substrates can fit into the active site of an enzyme. This is known as a lock and key model. Why lock and key? If the key doesn't fit into the lock, it can't open it. It's a bit like a jigsaw. It must fit in for it to digest. And by the way, this special region here is called the active site, which is where the binding occurs. Now, I'd like you guys to complete this. Pause the video, you've got 5 minutes. Off you go. Now I'm going to go over the answers now. Okay, so, enzymes are biological catalysts. They have a special region called the active site. The active site has a special shape which fits the substrate. When the enzyme and substrate collide, an enzyme-substrate complex forms, where the substrate binds with the enzyme's active site. An enzyme can only catalyze one reaction, as the substrate must fit into the active site for the reaction to take place. Well done for the students who got that right. Now very quickly, if you guys are interested in any aspects of science, this is a very good job that you can look into. But more on this when we look at bacteria. So here's the answers. I'll just wrote them down for you guys if you guys are struggling, girls are struggling to write them in time. Okay, now what I'd like you to do, this is your task. write down your favorite meal. It must contain a carbohydrate, a protein, and a fat. If I were to choose, that's me over there, I would choose a chicken cheese pasta. Why? Carbohydrates in the pasta, chicken protein, and cheese has got fat. So I'd like you to have a go at picking your own. Maybe you could do a lasagna. Maybe you could do a pizza. Maybe you could do fish and chips. Fish and chips. Fish, protein, chips have got carbohydrates and fats. Potato gives them the carbs. Fat it's made in the oil. Fantastic. What we're going to do is we're going to be finding out how this is broken down in our body. Now, first of all, the pasta, which is the carbohydrate source, will start digestion very early inside the salivary glands in the mouth. Amylase is an enzyme, so one of the key enzymes. Amylase is a carbohydrate type of enzyme, and this is one of the key enzymes that you need to know by the end of the lesson. And what that does, it's based in the salivary gland, and it turns starch into maltose. Maltose is a simple sugar. Okay, so amylase is also found in the salivary gland, the pancreas, and the small intestines. So already digestion is taking place. Okay, why does breaking things down matter? Why? Because our cells are very small, but the food products are very big. So we need to break down food products in order to pass by the cell membrane, because you need to understand some molecules, some food products are too big to pass through, so they need to be broken down. I think that's very important to understand. Now what I'd like you to do is put these in order. I'm going to be explaining to you the importance of peristalsis. Okay, let's go over the answers. So that's 1, 2 and 3. Now, what is peristalsis? Food is pushed through the body using special muscles in a process called peristalsis. Series of involuntary wave-like muscles contractions which move food along the digestive tract. Now it's important. Look, we're trying to get our food from one location to the other. So there has to be some sort of movement, and that's done by these involuntary muscles. Okay, so as you can see, circular muscles contract whilst the longitudinal muscles relax, and they are antagonistic, and that's the way it works. Okay, so that's helping the food down here. I've got a photo for you guys to understand as well. So circular muscles relax, longitudinal muscles will contract, and that will help the food move through, and that's called peristalsis. It's very important. You don't want your food to get stuck. The next thing, my chicken. The chicken in my lovely meal is broken down inside our stomach. Now, the stomach acid doesn't do the digestion. That's a misconception. The acid in the stomach creates an environment for the protease enzyme to work. It likes to work in an acidic environment. So it's very important you understand that concept. And proteins are broken down into amino acids. Okay, now my name is Mr. Amin. I teach my students, my name is Mr. Amin, so just add an O, amino acids. So that's the way my students remember. And protease is found in the stomach, the pancreas, and the small intestine. Very important to understand. The stomach also pummels the food with its muscular walls. That helps as well in the mechanical process, as well as peristalsis of the breakdown of food. So here we go, breaking down into amino acids, and amino acids are nice and small, can fit through. That's fantastic. Now the last thing is the lipids, the fats. How are they broken down? They're broken down by lipase, another type of enzyme, right? So lipase is found in the pancreas and small intestines, and that helps break down the lipids into fatty acids and glycerol, and that's how my lovely cheese is broken down. Now, a quick summary. So amylase, carbohydrates, starch, pasta, simple into simple sugars. So that's the enzyme, that's the product, and broken down into simple sugars. Protease is the enzyme that breaks down protein into amino acids. Lipase is the enzyme that breaks down lipids into fatty acids and glycerol. And that's the way my lovely meal would have been broken down. Now, some exam questions for you guys. Off you go. Okay, I'll go over the answers. You can pause the video, by the way, when you want to answer. Okay, what type of enzyme breaks down carbohydrates? It's carbohydrates. So, sorry, which type of enzyme breaks down carbohydrates? So it's carbohydrates breaks down carbohydrates. Which type of enzyme breaks down fats? Lipase. What type of enzyme breaks down protein? It's protease. And what type of enzyme is amylase? It's a carbohydrates type of enzyme. Well done for the students who got that right. Now, this is important. So, our food's being broken down. Where is it absorbed? Most of the absorption happens inside the small intestines. The small intestines are lined and packed with villi, which contain microvilli, which have a large surface area, and they absorb most of the nutrients. So a lot of the absorption happens here. Very important. They are jam-packed with blood capillaries as well, so they can do as much absorption as possible. Now, some more questions for you. You can pause the video now. Okay, let's go with the answers. What is an enzyme? Well, an enzyme, let me just see if I've got the answers. Right, okay, I'll read the answers. Why am I here? What is an enzyme? An enzyme is a biological catalyst which breaks down large molecules into smaller ones. A catalyst is something that speeds up a reaction without being used up. Proteases are digestive enzymes. Where in the digestive system are they found? In the stomach, small intestines. Describe the structure of an enzyme. I would use some of the keywords over here. So the structure of an enzyme, it's 3D. It contains an active site, and the structure, they're made out of protein, and the enzyme and the substrate must fit into each other perfectly in order to digest the substrate. Okay, now describe the process of peristalsis. That is an extension question for you. If you want to answer it, that's absolutely fine. Now, this is your main task for today. Write a paragraph explaining your ideas. You should include the different enzymes in the body, what these enzymes break down, where the smaller molecules are absorbed, how does a food substrate move inside the human body. This is typically a 6-mark question. I'll give you 6 minutes to answer this question. Okay, let's go over the answers. Now, if you were struggling with that task, I would have given you one of these. Now, this is a paragraph I've created. I've just taken out the keywords. So if you can answer this, it's like answering the major question. So enzymes are biological catalysts. Let me see one. I think I've got the answers. No, I haven't. Sorry. Okay, we'll go over it together. Enzymes are biological catalysts, which break down large molecules into smaller ones. This is important, so broken down food substrate can get into cells. There are 3 main types of enzymes. Amylase breaks down starch into simple sugars. Protease breaks down proteins into amino acids. And finally, lipase breaks down lipids into fatty acids and glycerol. Broken down substrate is absorbed by the small intestines, which have microvilli for increased surface area. Different muscles in our body help food move along our body using a process called peristalsis. Thank you very much. If you understood that concept, you're an absolute superstar. Now, what we're going to do in our next lesson, just so you can finish off the plenary, next lesson, we're going to be looking at how enzymes are affected by certain things, and how they're affected by temperature and substrate concentration, etc., and take this lesson more into Key Stage 4. Thank you very much for watching. Please like and subscribe, and peace.